Good afternoon. This is Dr. J.Guerra coming to you from VerEvMed and Authentic Biochemistry Studios in the Inland Pacific Northwest. Today we're going to do part number 2 on new neuroimmunoendocrine factors in major depressive disorder. And so let's get right to it. Today is the 23rd of October, 2019. I'm Dr. Daniel J.Guerra, and there is my Facebook page down there in the bottom. Here's the YouTube link for getting any of these lectures. They are free to the public. And here is also my Authentic Biochemistry link where I do my podcast. Today's going to be a video lecture, so it's going to be coming off of this feed, off the YouTube feed. So let's get started. All right. So first, let's do a little bit of a review where we would be finding potential nuclei and sites in the central nervous system where there has been research that suggests this association with disorders of the kind that we call depression. So these here are the anatomical locations of key brain regions. So this is a midline sagittal view, of course, of the brain, and it's an artist's depiction. And you see this various subsections, including the anterior cingulate cortex, which is labeled that, the amygdala, the hippocampus, the corpus callosum, which we talked about several times over the last year or so when I've talked about various kinds of neuropsychiatric disorders, including schizophrenia. The dorsomedial frontal cortex, you see these various regions here. You can just look at them while I'm labeling them off. The mid-cingulate cortex, right there. For example, the orbital frontal cortex, the pregenual anterior cingulate cortex, the posterior cingulate cortex, and the ventromedial frontal cortex. All of those have been associated with major depressive disorder from various research papers. I get this particular discussion from Journal Clinical Investigation 2009, and you can see the citation there. To give you an idea about potential anatomical regions in the central nervous system. So, abnormalities have been reported in the prefrontal cortex, basal ganglia, hippocampus, thalamus, cerebellum, and the temporal lobe. In individuals suffering from unipolar depression, the most reliable findings are in the prefrontal cortex, and again, the basal ganglia, and sometimes in the hippocampus and cerebellum. There is also the possibility of a relationship between hippocampal volumetric reductions, and as I mentioned last time, on white track and isotropy, all associated with the HPA axis. And this is all correlated with a glutamatergic theory of MDD, which is going to be different from the catecholamine version, right? Okay. Familial causes of unipolar and indeed bipolar depression show an abnormality in the prefrontal cortex, or PFC. Some studies have found altered volumetric measures of the prefrontal cortex, for example, reduction in the amount of tissue in the PFC of young unipolar depressed patients, and possibly a reduction in the size of a particular region of the medial prefrontal cortex in more adult patients. Postmortem studies have found a reduction in the number of glial cells. Now remember, those aren't neurons. Those are part of the major cell type in the central nervous system, which include frank glial cells, but also things like oligodendrocytes, astrocytes, and of course microglia, which are resident macrophages, all of which can play a role via the neuroimmuno pathway, and of course the white track association of the myelinated sheath around neurons are associated with oligodendrocytes, which actually aid in the process of laying down that myelin sheath, which is a sphingomyelin and galactosylceramide associated sphingolipids that coat neurons and give you the saltation process I talked about for those white tracks. So besides that, you also have extensive connections, of course, from the PFC to the amygdala. This is all part of the affective portion of the central nervous system. Nucleus accumbens, hypothalamus, and you have norepinephrine, 5-HT, that's 5-hydroxytryptamine or serotonin, and of course the dopamine pathways, all associated with the brain stem. Okay, so Functional imaging studies have also reported abnormalities in function. Increased activity may occur in some areas of the PFC, particularly the posterior orbital, the amygdala, and the anterior cingulate. Consistent with the literature that you can find, suggests that sadness and anxiety in normal subjects increases the activity of all those areas as well. So the increases in the ventral, orbital, and cingulate cortices may be related to emotions and or even obsessive ruminations. That's why sometimes there's a link between obsessive compulsive disorder. Okay. Again, these are broad strokes. These are anatomical features that have been associated either from scans, as we talked about last time, or post-mortem alterations in the actual brain nuclei that seem to have these either volumetric changes or there may be other disruptions, all of which could be associated with some kind of anisotropy. And this may map to alterations in neuroendocrine and in neurotransmitter biosynthesis, as well as reception and signal transduction cascades through those intracellular pathways that I've talked about quite a bit, such as the JAK-STAT pathway. So it's a very rough cartoon. Here's showing you what a neuron can look like, or a synapse between two neurons. You're going to have a lot of mitochondria associated with that. Why is actually a lot of energy to carry out neurotransmission. It's just showing you you have tryptophan going to 5-HT, going to 5-HIAA, which is a degradation product. There's serotonin itself, those red colored marks there, moving out of the presynaptic neuron to the postsynaptic neuron. There's a reuptake pathway where the any of these neurotransmitters can be picked back up. So not just 5-HT, which is serotonin, 5-hydroxytryptamine, but also norepinephrine, dopamine, acetylcholine, GABA, gamma-aminobutyric acid, that is, and glutamate and even glutamine. This is all in the postsynaptic neuron. This is the synaptic space, or the synapse, where this reuptake occurs. And so we all have heard about SSRIs and SNRIs, which of course are drugs which will allow for the reuptake of these neurotransmitters back to the presynaptic neuron so that they can be relocated to the postsynaptic neuron, right? And so therefore increase the biological activity of things like serotonin or norepinephrine. This is a major drug that's used for major depressive and unipolar disorder. And it's come under fire a lot after the STAR-D research paradigm, which basically showed that placebo often works as well as SSRIs and SNRIs when compared to when compared to the drug itself, and that there's a lot of remission that occurs. There's also a lot of plateauing effect of taking these drugs, these SSRIs and SNRIs, and that means that maybe this pathway needs to be looked at. And we talked about the glutamatergic pathway in previous VerEvMed and authentic biochemistry lectures that may be associated with various neuropsychiatric diseases, as well as unipolar depression. And I'm not going to really cover that today. We're going to really link into a potential comorbidity, which is associated directly with leptin, which is an adipokine, which is secreted from the visceral adipose, which can be elevated and can be desensitized. So you have leptin resistance that's associated with obese patients that also suffer from depression. So, again, the regional CNS neurotransmitter distribution here, I want you to understand the PFC, dopamine, serotonin, norepinephrine. The amygdala also, of course, is directly bathed with dopamine, serotonin, norepinephrine. The nucleus accumbens, serotonin, dopamine. Hypothalamus, serotonin, dopamine, norepinephrine. Striatum, serotonin, dopamine only. And the cerebellum, serotonin, norepinephrine only. So there are a lot of really well-done characteristics about neurotransmitter utilization, receptors, and biosynthesis for very specific regions of the central nervous system. Striatum is a convergence point for glutamatergic inputs from cortex and the thalamus, as well as dopaminergic afferents from the midbrain. It's also a source of direct and indirect pathways. So you have two parallel basal ganglia circuits that are critical for motor function and indeed for procedural learning. These all can be impacted in these neuropsychiatric disorders. Striatum is also associated with obsessive compulsive disorder, which I mentioned a minute ago, and also directly with addiction, because it's linked up to the dopaminergic pathway, the reward system, as it's called in layman's terms. Here's a paper published in 2015 from Neuropsychiatric Disease Treatment. The CNS anatomical regional specificity of that links cognition and mood are innervated with monoamine neurotransmission, including tryptophan derivative serotonin, and the tyrosine metabolites, these would be the catecholamines, norepinephrine and dopamine. Okay. Crosstalk among those neurotransmitters are both antagonistic and agonistic. Ligand activation, for example, of the 5-HT2A, that's the receptor, and the 5-HT2C inhibits postsynaptic norepinephrine and dopaminergic neurotransmission. See, so there's crosstalk there, that's what I'm saying. The D2-like receptors promote 5-HT neurotransmission, so that is agonistically potentiating the serotonin pathway. You also have presynaptic alpha-2 adrenocorticoreceptors and adrenoreceptors directly, which antagonize 5-HT signaling. And finally, you have postsynaptic 5-HT receptors, such as the 5-HT1A, the 5-HT3, the 5-HT7, which all modulate not only via the serotonin, right, GABA interneurons, which subsequently alter serotonin, acetylcholine, norepinephrine, dopamine, and glutamate neurotransmission. So it's much more complicated than simply discussing the reuptake of something like serotonin. Far more complicated than just talking about concentration gradients across the synapse. Right? And this has been well known for a long time. Hence resistance of a lot of these drugs for people that suffer from various neuropsychiatric conditions. So the MT drugs target the monoamine pathways by increasing neurotransmitter synaptic residence time. Okay, I'm sure all of you are aware of this, clinicians in particular. So you have citalopram, escitalopram, reboxetine, sertraline. Those are all SSRIs. SNRIs include duloxetine and venlafaxine. The NDRIs, can include the very heavily used drug these days, bupropion. You also have tricyclic antidepressants. It's kind of old school. Amitriptyline, desipramine, and imipramine. And you have alpha-2 adrenergic antagonists, and that would be mirtazapine. Okay? These are all drugs that have been used over time, over the last really decades, to treat depression, particularly in adults. Okay? All right. So, take a look at this now. This is some newer material. Trends in Molecular Medicine, published in 2016. There's your citation. Okay, so in instances of hyperleptinemia, such as diet-induced obesity, now this is going to be a subclinical animal model, diet-induced obesity, and inflammation, leptin appears to promote or enhance the inflammatory response mechanisms of immune cells. So, what does leptin do? It can be a licensor of immune function. So it can give you a microbiocidal response, leukotriene production, lipid body formation, phagocytosis of pathogens. So this is a licensing of immune function. You also have a response to inflammatory stimuli like cytokines and chemokines. And you also get maturation potential increases because you're using chemokine receptors, the major histocompatibility 2, which is an antigen-presenting protein that's on the surface of these cells. And you also have the CCR7, and you also have CD80, which is a co-adapter molecule, which is always on these stimulatory cell lines. You also have an enhancer of immune function. These are going to be the toll-like receptors, right? So you have chemotaxis towards ataxin degranulation, and then the Th2, that is the T lymphocyte 2 responses, which are cytokine, which we're going to generate cytokines, and that's going to be generated via production in the basophils. So you're going to have an induction of the ROR gamma T receptor system, which is going to be essentially nuclear transcription factor mediated expression of such cytokines as the cytokine Th17 pathway, and that's going to also induce and trigger the activation of Th17. That's T helper cell 17 because they're producing cytokines of the 17 particular molecular structure. Again, you have a surface expression of the toll-like receptors. This is going to be linked more to the innate immune response. You're going to have an interferon gamma producing phenotype. And that's going to polarize to M2 macrophages. M2 are the quiescent macrophages. So you have chemotaxis towards leptin, resulting in increased infiltration to sites of infection and inflammation of all of these particular cell types here. Okay. So you also have an inhibitor of immune function. So you have a reduced cytotoxicity and interferon gamma secretion after extended leptin stimulation, such as in leptin resistance, okay? And this has to do with people that are chronically obese or have high visceral fat. You also have suppression of the Treg differentiation because of leptin. And so if you suppress Tregs, that means you don't have suppression of the Th1, Th2, and Th17 T lymphocytes, which means that you have a pro-inflammatory response, right? Likewise, and these are all the different cell types here. They're all just, so monocytes, macrophages are involved, dendritic cells are involved. These are the ones that have this MMH2 class, receptor-mediated induction into the T cell lineage, which again then induce B cells, produce immunoglobulins, okay, if you remember from your immunology courses. Basophils are involved, eosinophils are involved. We talked about that. This can be an allergic response, actually. Neutrophils, mast cells, natural killer cells, CD4 T lymphocytes, which is essentially precursor to all the T lymphocytes, including Th1, Th2, Th17, and of course the Tregs. And those are the two cells down there. So, about leptin. Low leptin, malnutrition, leptin and leptin receptor defects are involved. You have a decrease in functional abilities. Normal leptin signaling, remember this is an adipokine, cells are primed and ready to respond to the stimulus, so they're in the perfect zone. And then elevated leptins, what you get in obesity, you get inflammation because of this licensing of immune function, enhancing of immune function, and shutting down of T regulatory cells, And what you basically have are cells that are activated with enhanced functional abilities. Okay, so this is all at the extreme case where you start to get leptin resistance. So here we're talking about the comorbidity of obesity linked to the immune response, which links back to the neuroendocrine system and neurotransmitter system, which can give you then a comorbidity of obesity as associated with leptin, the adipokine leptin, to major depressive unipolar disorder, for example. That's what we're talking about here. All right. Paper published in Frontiers in Neuroscience very recently, 2019. There's your citation. It was actually published in July. This is October. Paper is Relationship Between White Matter Integrity and Plasma Leptin Levels, White Matter Integrity and Plasma Leptin Levels in Drug-Naive and Medicated Patients. This is a clinical trial with major depressive disorder. So remember last time we talked about white matter tracks. Now we're including white matter tract anisotropy with leptin expression. So we're putting in this next factor. This is how I do it in my how I teach in my lectures at university, and also how I give open lectures when I'm talking neuropsychiatric societies. And now I'm also doing this in VerEvMed lectures and also in authentic biochemistry. I try to include the basic scientific literature and come up with a what I call a die-event ontology of what we know in three-dimensional space-time about given topics in biomedicine. Here we're keying in on neuropsychiatry. In the past I've done, of course, all other aspects of biomedicine, including all things like chronic kidney disease, hepatocellular carcinoma, heart disease, aging, things like that. Okay, so just a little bit of a anecdotal affront to your senses associated with what I'm doing in VerEvMed. Okay, so let's go back to this paper. White matter tracts are implicated in the course of MDD since various anatomical and functional abnormalities of that white matter, remember that's the myelinated tracts in the brain, seem to correlate to severity of the disease. Since the MRI technique diffusion tensor imaging, DTI, which I talked about last time, uniquely characterizes and follows water movement in white matter fibers, what's called the fractional anisotropy, FA-DTI-derived diffusivity measure, is therefore employed in these clinical trials to track the directionality of water diffusion and thus reports on the locus and strength of white matter tracks. Now, why is water? Well, you're following the water, but understand what's dissolved in the water is what you care about. What's dissolved in the water? Neurotransmitters, okay? And of course, sugars and any other kind of bioenergetic compounds, and even bioactive amines that are not neurotransmitters, and even amino acids that can be converted to carboxylic acids to run through the TCA cycle and generate ATP, because you need a lot of ATP to do neurotransmission. So you know all this if you follow my biochemistry lectures, and also you would have some ancient memory of this when you took biochemistry from someone like me when you were in medical school. Okay, so in many DTI studies, the FA, remember that's fractional anisotropy, are found to be lower for patients with MDD compared to healthy controls in different white matter regions, such as those regions we just looked at: thalamus, frontal lobe, the uncinate fasciculus, the corpus callosum, the cerebellum, the parietal lobe, and the superior longitudinal. Okay. There is a link between, okay, this is now prolegomena introduction to this paper, prolegomena, right? There's a link between obesity and, since leptin is produced in adipose tissue via tight fat mass ordered transcriptional regulation, okay, the idea that leptin could affect the brain towards MDD, therefore, is logical. That's the important thing I like to do in all of my lectures. Does it follow the square of opposition? Can we find logical arguments for thinking that leptin could be associated with a neuropsychiatric disorder? And there, right there, describes it. Because we know that leptin acts as an adipokine, which basically signals through cytokine receptors and then subsequently subcellularly the JAK-STAT pathway, leading to alterations in gene expression. And that gene expression can include things like the production of neurotransmitters or, in fact, pro-inflammatory cytokines. Hence, you get the link between a morbidity such as obesity and major depressive disorder. Okay, so it's logical. Doesn't mean that just because something's logical doesn't mean that the argument is sound, right? Doesn't mean that all the premises are true and therefore the conclusion follows from those true premises, right? You have to then find empirically the evidence that this indeed is the case. And that's what papers like this are trying to do. All right. So let's go back and to discuss this a little bit more. This comes from the International Journal of Molecular Science. Again, I use a lot of papers when I talk. Adipocytes can produce complement factors, cytokines, and chemokines, and of course, adipokines, such as leptin, one we're looking at today. Now, adipokines are associated with insulin sensitivity. So, in fact, there's crosstalk between insulin and leptin. And I've talked about this in previous authentic biochemistry lectures, so I'm not going to bore you with those implications. I just want you to know that insulin and adipokines like leptin crosstalk. Cardiovascular disease is associated with adipokines. Arthritis, such as rheumatoid arthritis and osteoarthritis, which of course are inflammatory diseases. And then therefore, big time, obesity. Okay, so let's take a look at this now. Obesity, what does it have that's going on in terms of, you have insulin resistance in obesogenic systems in humans, right? That means that you produce insulin, like type 2 diabetes, but the insulin reception is corrupted. So that means there's a high levels of circulating glucose, and it also means there's high levels of circulating lipoproteins from and to the adipose from the liver. And that can induce atherosclerotic plaques, which can lead to heart disease, which can also lead, because of pro-inflammation in subcellular regions and other organ beds and in other angiosystems, to oncogenic events, which can lead to cancers, right? All of that can occur because of insulin resistance associated with adipose. And what is adipose doing, especially in the obesogenic? Producing free fatty acids, increasing amount of free fatty acids, which disrupt membranes. Produces a lot of leptin, right, the adipokine. Tumor necrosis factor alpha, pro-inflammatory cytokine, IL-6, pro-inflammatory cytokine, and MCP1, which is a growth factor, which helps enhance all of these activities. Now, how does that compare to, for example, people who are 100 years old? What happens from their adipose tissue, of course, is usually a lot less of it than obese people. So some adiponectin is produced, no leptin though. Some lipoprotein lipase is produced. That's good because it's removing triacylglycerol, laden fatty acid and triacylglycerol from circulating VLDL and LDL, right? But you get a tanking of free fatty acids, and you get a tanking of leptin. And you're very insulin sensitive, which is very good, right? Low glucose levels. And what about lipodystrophy? Here you get ectopic lipid deposition, such as in the cardiovascular bed, or let's say in any place where you have a lot of blood vessel vasculature, which can then enhance a pro-inflammatory response, which then can enhance, for example, different kinds of cancers throughout the body, even in the central nervous system. And I've talked about this in previous talks. So here you have ectopic lipid deposition all over the place. You have free fatty acids accumulating. Free fatty acids are always a bad thing because free fatty acids can cause membrane disruption. Okay? Adiponectin tanks and leptin tanks here too, but you still get insulin resistance. So with lipodystrophy and with obesity, which are usually linked because you have dyslipidemia in obese humans, all of them lead to, here, in very direct consequences of having insulin resistance, which means high circulating glucose, which means glycated hemoglobin, so you get diagnosed with type 2 diabetes, and you have a whole problem with dyslipidemia and deposition of lipid in various vascular beds where it shouldn't be, and the diseases the top diseases that cause mortality and morbidity in humans then are linked coherently to having obesogenic lipodystrophic systems, right? And this is just in comparison to elderly people who don't have obesity or lipodystrophy, right? And why do they live that long? Possibly because of that, see? All right. No, let's go back to leptin itself. Leptin, our favorite adipokine. At least it seems that way right now. Leptin helps regulate synthesis of thyroid hormones. It increases blood pressure. It decreases glucose-stimulated insulin secretion. It increases heart rate. It activates a series of immune cells. We already saw that 3 slides ago. Regulates appetite. Remember that. Okay, it's an anorexigenic system, so it decreases the appetite. Okay, so rather than orexigenic, which just causes a gustatory response that makes you want to take in food, an anorexic response causes you to not take in food. That's what leptin is used for, the adipose to the brain, signaling plenty of adipose tissue, stop feeding. So you can imagine if you become leptin resistant, plenty of adipose, plenty of leptin produced, plenty of all this activation of the immune response and increasing blood pressure, such as in various kinds of metabolic diseases, such as metabolic syndrome, right? And in type 2 diabetes, too often associated with obesity, is hypertension. But at the same time, what else you're doing is you're dysregulating appetite. So obese people can eat and eat and eat and not feel full because the leptin response is desensitized, right? It's resistant. There's also regulation of the menstrual cycle, which I plan on doing a whole series of lectures on. Menopause, a major cause of disruption, dyslipidemia, and can lead to leptin resistance. And also regulates bone mass because, in fact, oligodendrocytes that are found in bone mass also lead to deficiencies in the production of mineralization of bone cells, which is similar to what happens to adipocytes, only there is triacylglycerol, not mineralization, which is corrupted. All right. So normal leptin, what happens here? Okay, this is supposed to be a diagram of the brain. So normal fat mass, normal leptin signaling, brain acts on the signal, you have an anorexic control. Normal appetite, increased metabolism, good weight control. Okay. Low leptin, brain acts on the signal. That's because of reduced fat mass, right? So on lean body mass people, reduced leptin signal, body's in a starvation mode if this is severe, increased appetite. So now you have an orexigenic response. Orexigenic means inducing appetite. Decreased metabolism, so you don't want to be wasting the calories that are already deficient. Now, leptin resistance, watch this. The brain only receives part of the signal. There's some kind of corruption in this, so you don't get good leptin reception. Therefore, signal transduction cascades leading to enhanced nuanced gene expression, because that's all because of excessive fat mass, such as in obese people. Response is increased appetite, because you're resistant to the leptin signaling. Decreased metabolism, because it's still doing the same thing. Leptin isn't working correctly, so it looks like leptin deficiency, you see. Does this sound like insulin resistance? You bet. And you also get weight gain, because people keep on eating and eating. Increased risk of metabolic syndrome, type 2 diabetes, and cardiovascular disease and cancer, which are not listed here, but I'm just adding because I know that's true because I read the literature. All right. Paper, Nature Metabolism, published very recently, 2019. There is your citation. What is this paper going to tell us? It tells you that the first law of thermodynamics, right, energy conservation, energy within a system has to be conserved, and that any change must result from either a change in the energy input or output into that or out of that system, right? That's what that first law of thermodynamics states. So chemical thermodynamics. Thus, among mammals in which energy is stored primarily in energy-rich triacylglycerol, it should be called triglycerides, you have 9 kilocalories per gram, remember, found in adipose tissue. Any change in weight, which is then associated directly with fat mass, must necessarily be a result of changes in energy input, food intake, or energy expenditure, right, such as exercise. For weight to remain stable in a living organism, energy input thus must be precisely balanced against output over long periods of time. That's why we're talking about first law of thermodynamics. Of course, full stop. When food is readily available, then, adult humans maintain a remarkably stable weight while consuming approximately 1 million kilocalories each year. Collectively, the findings have long suggested the existence of a biological mechanism indexing food intake to energy expenditure, because you have to have a maintenance of the stability of weight. This is what happens through most of our adult life, unless we become obesogenic, right? Adipose tissue mass is also maintained at a specific level, a pretty narrow range, actually. Maintaining the stability of energy stored in adipose tissue is also essential for survival, because lower levels of body fat have deleterious consequences by increasing the risk of starvation, right, during periods of food insecurity or famine, as well as by decreasing fertility in younger people and decreases in immune function throughout the lifespan. In contrast, higher adipose tissue is also a problem, right, because it increases the risk of predation, right? High body mass, slow moving, you can be preyed upon. It has thus been postulated that nutritional state, probably from a fat-derived signal, is sensed to maintain homeostasis of adipose mass. However, the identification of the molecular components of that putative system were elusive, owing in part to the intrinsic difficulty in applying biochemical methods to identify signals that are physiologically regulating food intake or energy expenditure. That's because most people are not lipid biochemists. Difficult to do lipid biochemistry because you have to use organic solvents. Many biochemists don't like chloroform, methanol, things that we have to use every day in the lipid laboratory. It's further compounded by the ability of many factors to decrease food intake. You also need petroleum ether in a lipid biochemistry laboratory, which is a lot of people will definitely not want to use. Anyway, by non-specifically eliciting an aversive, non-physiological response, including nausea and other unpleasant sensations, which can be linked to overeating, right? Yeah. Okay. So that gives you, again, a kind of a understanding of what this paper is trying to talk about. All right. So we already took a look at this. I'm just giving you sort of a further background in leptin, right? So leptin works through something called the leptin receptor, a protein that's found in the membrane. Remember I told you dimerizes, works through the JAK-STAT pathway. When I say, remember I told you, that's of course all of you are very, very committed, confessional type of listeners to Authentic Biochemistry, my podcast, and therefore you know all about leptin receptor. So I don't need to bother you with continuing that long paradigmatic pathway. I will tell you that there are central actions, hormonal regulation, food intake, energy expenditure. I'll also tell you that bone and cartilage also are regulated by leptin, vascular function, such as atherosclerosis, angiogenesis, laying down new vascular beds, and even oxidative stress are linked to leptin. Talked all about that in the past. reproduction, such as placental function, trophoblast invasion, fetal function, gonadotrophins, and gonadal hormones are also regulated by leptin. Other targets, kidney, bowel, pancreas, and muscle. Bowel, particularly when you think about inflammatory bowel disease. Again, I talked about that in a previous Authentic Biochemistry and VerEvMed video lecture. And then there's this entirely huge area, innate and adaptive immune response, cytokine induction, chemotaxis, natural killer cytotoxicity, macrophage activation, or going from M1 to M2, which basically shuts down the macrophage pro-inflammatory response, dendritic cell activation, going through the MHC2, which then turns on T cells, okay? And that then links to Th1, Th2, and stimulation, inhibition of Th2, naive T cell proliferation. All right. I talked to you before about leptin binding to its receptor. There it is. There's the JAK-STAT pathway, the SOX3 pathway. There's the phosphatase, which then starts shutting it off because it removes these phosphates from these tyrosine residues on this dimerized leptin receptor. Leptin binds its receptor, binding of the long form. That's the guy here, long form. There's a short form and a long form. It has to do with splice variants, of course. You turn on a JAK pathway, which is Janus kinase pathway. Autophosphorylation of these tyrosine residues. Phosphorylates all those up to the cytoplasmic domain of the LEPR on those particular canonical tyrosines. Each one functioning as a docking site for cytoplasmic adapters. All of these, like the SHIP, the JAK2, the STAT3 pathway, all are docking onto these phosphorylation sites. Now it's occurred because of the JAK kinase functioning, because of leptin binding and signaling through this receptor. It's much like a cytokine receptor. It's an adipokine receptor, much like it though. All right. So anyways, yeah, you have a STAT pathway, which is the signaling transducer and activator transcription, STAT, okay? Because that's going to go right to the nucleus, and cause nascent gene expression via chromatin remodeling and transcription factor mediated response to specific genes, such as, for example, pro-inflammatory cytokines, right? Yeah. Additionally, leptin induces activation of that SHIP2, which recruits the adapter protein GRAB, which prompts the activation of the RAS, RAP, MAPK. These are all sometimes recruited and dysregulated in oncogenic events, such as many forms of cancer. Breast cancer was one I covered recently. Leptin also mediates, and breast cancer associated with obesity in females, right? Particularly around perimenopausal women. Leptin also mediates the PI3, the phosphatidylmalonyl kinase pathway, which is associated with AKT, which is another kinase. Activation via insulin receptor substrate, that's IRS1-2, and a protein tyrosine phosphatase that shuts everything down. So I keep on saying this to people that want to learn more about biochemistry. Every time a receptor binds, every time a ligand binds with its receptor, and it induces a forward-moving cascade, such as the expression of a gene, okay, which is basically what's going on here. It immediately turns on, not later, immediately turns on a pathway, and that's what this PTP1B is, that starts stripping the phosphate off and frequency and amplitude modulating, turning down the same induction that just got caused by ligand binding. So as ligand binding induces a response, a proactive response, it also induces early on an inhibition of that response. And that's why when you take a drug that's working to, say, alter leptin binding or leptin receptor mediated, or if it's cytokine mediated, such as knocking down pro-inflammatory responses, or let's say it's an SSRI or SNRI, right? It goes completely around these very tightly regulated pathways so that you don't have feedback regulatory control, which means that when you take a pharmaceutical or you just take higher levels of even a dietary compound, which is bioactive, such as a particular kind of fatty acid with, say, a specific geometrical positional isomer, N-3 versus N-6, right, a polyunsaturated fatty acid, very long chain polyunsaturated fatty acids, that can completely disrupt receptors. Something you don't think about when you think about when you prescribe drugs, but which indeed does happen. And that's why you get drug resistance so easily. And that's also how addiction works, by the way, just so you know. So, a clue to the identity of factors that regulate energy balance is provided by the identification of the recessive mouse, the OB mutation, and a subsequent identification of the DB spontaneous mutation, DB for diabetes, of course, both of which cause mice to develop massive obesity and show identical phenotypes of the same genetic background. So you probably saw this if you were looking at the cover of Science back in the late 1990s when leptin was first discovered. That's what led to this. Similar phenotype also develops in genetically obese rats carrying the FA mutation, fatty acids, what the FA stands for. These fully penetrant recessive, these are recessive mutations, cause extreme obesity and insulin-resistant diabetes as part of a complex syndrome, which includes a broad set of unusual abnormalities which somehow seem to be linked. So with obese people, you get a problem with infertility, immune alterations, usually pro-inflammation, and you also get hypothermia, which is interesting. And I talked about this in the past, and someday I'll talk all about thermal regulation in humans because it's fun to discuss, especially as winter comes on. The extreme obesity of all those mutants resembles that in animals with lesions in what has been previously mapped to the ventromedial hypothalamus, or VMH. Sorry, I didn't close that parenthesis. Animals with hypothalamic lesions, in fact, overproduce a blood-borne signal that normally decreases weight, but because of the lesion, they can't respond. So the lesion is in the production, the lesion is in the reception. Of course, that's what receptors are in the central nervous system. The lesion was further suggested to lead to a compensatory increase in the level of humoral factor that's found in the blood, right? That results in weight loss in a paired animal with normal hypothalamus. Okay, so you're looking at hypothalamic lesions, you're looking at something secreted from the visceral fat, and you're looking at all of these possible diseases, comorbidities, that are linked to other ablation of some region of the hypothalamus or something that's causing an inhibition of signaling to these other mutations, which were found out later to be leptin. In fact, OB mice are obese because they don't produce the hormone. Hormone was leptin, the adipocrine leptin, that regulates food intake and body weight. DB mice and FA rats are obese because they don't express the receptor for that factor. Okay, so, mystery opened, unbound, right? And the receptor is expressed in the hypothalamus. Okay, makes perfect sense when you start taking all the papers and taking it all in concrete, all that evidence, to do the epistemic movement and start generating a hypothesis, a hypothesis to think about how this could be occurring. You have to have a ligand binding to a receptor. The receptor has to mediate some signaling transduction cascade to cause gene expression. A disruption at the beginning or the end of that pathway, or anywhere in between, then will give you all those different phenotypes, which looks like a lot of them are obese. So isn't that interesting? That's the way biochemists think. It's the way we think. Now, finally, back to this frontiers of neuroscience. Is leptin linked to MDD? Okay, that's what we want to know. Is leptin linked to major depressive disorder? That's what this paper is going to talk about. Now, you have the leptin primer. You already knew about the major depressive disorder primer because I've talked about it before, but now you got both together. Now we can put them together. Okay, we're finished with the introduction. Now we're into the actual transmission of all this information into new knowledge. So hopefully, right? If we can find that things are true, and we can find out that they can actually be demonstrated, right? So true functional beliefs. White matter tracks are linked to major depressive disorder. We know that. Scanning interrogation of white matter includes MRI diffusion tensor imaging. I talked about DTI. Tracks water movement. I already said that. Fractional anisotropy. Anisotropy is a DTI-derived diffusivity measure, which can reflect the directionality of water diffusion. As I've said, it's used to indicate the location and strength of white matter fibers. That's why you use this technique. I want to make sure that when you learn from me in these lectures that you know that this is actually experiments are done. And if you want to know how experiments are done, you need to know what's being employed, right? What are the laboratory techniques that must be working correctly and have been vetted to be able to measure these things like anisotropy. So I'm giving you a little bit more detail than you might get just by glossing over materials and methods. In many DTI studies, FA values, remember that's functional anisotropy, not fatty acid, functional or fractional anisotropy. Actually, it can be functional or fractional there. Are found to be lower for patients with MDD compared to healthy individuals in different white matter regions, and I told you this before. Thalamus, frontal lobe, corpus callosum, cerebellum. Relationship between obesity and depression suggests a potential for leptin involvement because of this hypothesis being generated, right? If leptin dysregulation is specifically associated with MDD, then depressive symptoms need to be associated either with elevated plasma leptin or diminished leptin levels compared with non-MDD control patients. Either way, right? Both have been reported, thus we have an apparent antinomy, which means you have S equals P, S does not equal P. So because of the rule of logic, an antinomy, which means against the law, against nomos, the law in Latin, so you have A and not A. So you're allowing for the excluded middle term, Aristotelian categorical logic. So how can this work, right? You have an antinomy, right? Or you have an apparent antinomy. Well, not really, right? Blood leptin is transported into the cerebrospinal fluid, then into the hypothalamus across the blood-brain barrier. We've already studied this through various receptor isoforms, the OBR and the OBA through F domains. These are different splice variants and different expressed forms of the leptin receptor. So right away then you can see where there can be a variation in the signal. If there's a variation in the signal, then you don't need to worry about allowing for the excluded middle term, right? Because if you think about square of opposition, it's not all S or P or no S or P. It could be some S or P or some S are not P, thus completing the square of opposition, thus not allowing for contradictions, just contrary statements. Right? That's why it's good to know how to use categorical logic when you're thinking about how these experiments are designed and whether or not the results you get from them can be turned into perhaps evidence, and then after vetting the evidence and looking at the primary literature again, whether or not the corpus of the literature will agree or disagree with the current research paper. It's always important. It takes a lot of time to do this, and most people don't have the time, and that's my job to let you know how it's done and to do it for you if you care to ask those questions or any questions in biomedicine. Now, continuing. Remember that the hypothalamic leptin reception is positioned as a rheostat, okay, for visceral depot fat mediated behavioral control. So right away we know that leptin controls behavior. What behavior? Anorexic versus orexigenic. Okay, that's the behavior. The appetitive feeding, right? Leptin shuts down because it controls the neuropeptide Y and the agouti-related protein and the POMC pathway, right? And because it controls that in the brain, it controls satiety and the feelings of satiety, which can include nausea when you continue to eat. These are behavioral responses, clinical behavioral responses. So now you begin to see how this can link to neuropsychiatric states, right? Because there are behavioral responses which have to do with the simple aspect of appetite because you're a heterotrophic organism. Continuing. Remember, the hypothalamic leptin reception is positioned as a rheostat for visceral depot fat mediated behavioral control over the orexigenic anorexigenic axis and therefore entirely homeostatic bioenergetics. You use glucose, you use fatty acid for the production of ATP and which organ systems. Gets all the way down to that level. Okay. Again, it has to do with dyslipidemia because lipid is the true chemorheostat in the body. The visceral fat dictates whether or not the body has enough carbon to exist without taking in more food. And that ultimately also relates to all the essentials in the diet, such as the essential vitamins, metals, as well as essential amino acids and essential fatty acids. Because it's all linked through metabolism, TCA cycle in particular, because of transamination reactions and the utilization of the carboxylic acids from those amino acids after the stripping of the amino group, ultimately through the urea cycle, but recycling those amino groups and then transamination reactions, mammalian aspartate shuttle across the inner membrane of the mitochondria, regulating gluconeogenesis, glycolysis, fatty acid synthesis, fatty acid degradation, cholesterologenesis. All those things in metabolism, which I love to get into, and which, of course, the body is always doing autonomously as it's regulating the feeding. All of that is not described in this paper, but that's what you get when you understand the biochemical paradigms, right? You see how it comes together. And that's really important. Otherwise, papers like this give you a very unilocular event, ontology, which doesn't necessarily generate evidence that can lead even through to conclusions that the paper seems to be deriving. Kind of the case of what happens here in this paper. So besides the hypothalamus, leptin-responsive hippocampus typically associates with epistemic functions like learning and memory, and even the cortex, thalamus, striatum, midbrain, and other CNS nuclei respond to leptin. So it's not just the hypothalamus, okay? That whole thalamic arc. Obese patients are often hyperleptinemic. They have high levels of circulating leptin, and therefore they're leptin resistant, just like hyperinsulinemic type 2 diabetics are insulin resistant. Very similar paradigm. Right? Obesity was found to also be associated with reduced white matter integrity. We talked about that last time, right here at VerEvMed lectures, particularly in the genius, splenium, and fornix, where these white matter tract alterations map onto cognitive deficits. Yep. Despite growing evidences for white matter and leptin level abnormalities in major depressive disorder patients, there are no published studies examining their relationship until now. 2019, July. Perhaps hyperleptinemia, that was when the paper was published, I know it's October now. Perhaps hyperleptinemia is linked to altered white matter integrity in major depressive disorder patients. So a voxel-based analysis was performed, right? So now we're looking at the density using voxels, right? Using the MRI scanning data and the DTI associated with functional anisotropy and fractional anisotropy, functional fractional anisotropy, to be able to link now these leptin-mediated responses directly to voxel coordinates, right? They're going to be looking directly at the white matter. So you're linking two entire areas. You're looking at obesity-associated, major depressive disorder. And rather than being a period and say full stop, I want a semicolon there. All right. Materials and methods for this paper are the following. You start off with 140 participants. Okay? Not a lot of people, right? Not a lot of subjects. 40 are drug naive MDD, 40 are medicated MDD, and you have 60 healthy controls. They're relatively young, 18 to 49, and they're recruited from an outpatient department of psychiatry in Shenyang, China, and it was between 2013 and 2017. You had 17-item Hamilton Depression Rating Scale was used, the HAM-17, and the Hamilton Anxiety Scale was also used, HAMA, to assess the symptoms of all the participants. And obesity was tested by BMI. It's not always the best way to look at obesity, but okay, standard technique. MRI data was acquired using a 3.0 T-MR scanner, and diffusion tensor imaging data was acquired using a spin echo planar imaging sequence. Now, I've looked through this, and this is pretty standard technique. Okay. Some of the imaging data can be manipulated different ways depending on the paper that I've looked at. But the rest of this looks like the instrument being used looks like a typical high-tech, 21st century instrument being used. The diffusion sensitizing gradients were applied at 25 non-collinear directions, and the B on that was 1,000 seconds per millimeter squared. That's also standard. Okay. Together with an axial acquisition of diffusion weighting, B equaling 0. Scan parameters were repetition time, the TR was 17,000 millisecond, echo time and TE was 85.4 milliseconds. Standard imaging matrix is 120 by 120. This is the voxels, and that's the field of view, 120 by 120, very common. That's coming to a 24 by 24 centimeter space then that's spread out into that. You have 65 contiguous slices of 2 millimeter and no gap, with DTI using Panda software. Panda software is standard technique too. Okay. The images of diffusion matrix were transformed from native space to standard Montreal Neurological Institute using spatial normalization with voxel size again of about 2 millimeter cubed. Again, standard techniques. Okay. The fractional anisotropy value, therefore, was finally calculated using default program parameters, where the FA image of each subject for nonlinearity registered with the fMRI B58, which is basically the FA template, and the FA images were smooth with a 6 millimeter tail with full width at half maximal Gaussian filter. I've looked into this. These are very standard techniques. I'm just giving you the detail for those of you who do MRI. Immunoprecipitation assays for serum leptin, 5 milliliters of venous blood level, about 5 mils of venous blood were collected from all the participants between about 10 a.m. and 3 p.m. I don't know that time really matters. I would guess that'd be postprandial though, but lunch is right in the middle there, so who knows. The blood was spun down at 2000, at 10 minutes was stored at -80. Okay, leptin's stable at -80. I don't mind that. I would like to have seen maybe a protease inhibitor in there, and I assume they did put that in the cocktail. Measurements using a human pre-mixed multi-analyte kit with a human magnetic Luminex assay, so using magnetic bead assay to pull it down, right, using an immune precipitation technique. Magnetically labeled using a human magnetic premixed microparticle cocktail of antibodies. So they have monoclonal antibody, and this is basically a competition assay. Okay. All right. So here's some of the demographics. Again, I think you should look at this when you read a paper. Male to female, a lot more females in here, okay, with the drug naive, but the same ratio for the medicated. But look at here, in the control, it's almost equivalent male to female. So there's a little bit of problem here, but you can do the statistical pairwise comparison or PCA. But okay, they did the statistics and they find out their p-value there is reasonable, okay, related to gender, age, all pretty tight, around 30 years old, right? About smack dab in the middle of millennial. Again, okay, p-value. Education in years, very similar, right? Something post-secondary education. BMI, look at that low BMI. Drug naive, medicated, HC. Now, you just don't see that. This was done in the United States. That BMI would probably be at least 25, right? For just doing a cross-section of MDD people at that age group. Much leaner people in China. Okay. Clinical characteristics, disease duration, about 20 months for the drug naive, much more for the medicated because they've obviously been diagnosed and been on such as an SSRI for a while. So there's 37 months. And of course, healthy controls are not taking, there's no disease. HAMA scores, 15 on drug naive. Looks like they have a little bit of a decrease in medicated. You'd love to have seen a lot better value there, but I'm sorry, there wasn't. And of course, the healthy controls have a very low HAMA. The HAM-D, 17.3, 15.3. The drugs don't seem to be having too good of effect there. Again, way low, you would hope, with healthy controls. Leptin levels, okay? Here you go, 9,816, 5,047. That's about half as much for the medicated people. And then the healthy control is somewhere around the same as the medicated. It's kind of an interesting data point to me. The GI fractional anisotropy value, this is the white matter tract data, 0.26, 0.27, not much of a difference, okay? And again, with the healthy control, about the same, okay? With a p-value essentially not even need to be measured. So again, you've got people that are definitely suffering from major depressive disorder with these two scales, the Hamilton Anxiety Scale, the Hamilton Depression Scale, you see those two, delimiting numbers. This is all done with an ANOVA analysis of variance, doing a sample t-test, which is fine statistics as far as I'm concerned. All right, here's some data. So now you're looking at fractional anisotropy. You're looking at some of this coloration here has to do with showing you abnormalities. You're looking at the left thalamus, and it's corrected with the voxelate, and the p-value on this is 0.01, which is really good. And B is the post hoc experiments. You're looking at the data, and that's what these histograms are doing, just measuring this hot index here. And you're seeing that, what? You're seeing the drug naive, right, and the treated both have lower FA scores, even though there's pretty high standard deviation here, with the healthy control. So this tells you, because this was done quantitatively and it shows significance, here's the significance, healthy control to the naive, and then healthy control to the drug treated. There's a statistical difference, a major statistical difference here with the naive, very small statistical difference here, no difference between the medicated and the non-medicated, which is kind of interesting to me. So the white track differences, that's what this FA is measuring, between the drug-treated and the non-drug-treated depressed people, those white tract differences basically are negligible. The only differences are between the healthy control and either the drug-treated or the drug-naive. Higher leptin, lower with the fractional anisotropy, with the naive MDD having the most defined inverse relationship with hyperleptinemia, yielding the most aberrant white tracts. That's what you're showing here. The FA value lower, way out here with the drug naive, as the people out here, and there's a leptin level, right? So much higher leptin level associated with much lower FA, which suggests that you have more white matter disruption with higher levels of leptin that may be linked to having a naive major depressive disorder, drug-naive MDD patients. That's what this data shows you. All right. Analysis. Okay. So moving on. Drug-naive MDD patients presented with the most severe alteration of the dorsal medial thalamic white matter integrity, what we just saw, that was the data you just saw, evidenced by reduced fractional anisotropy and correlated significantly higher serum leptin. Okay. Diffusion tensor-derived directional water diffusivities are potential markers. That's what they're trying to argue here. It's been argued before. They say detect and differentiate axon and myelin injury. Therefore, reduction of that FA value is thought to be reflecting the reduction in the white matter organization. Direct readout, reduction of the axonal density, and a reduction of myelination where the thalamus may play a critical role actually in the pathophysiology of depression. You have to buy all of those premises. And previous papers have tried to argue for that. I won't go through it now. The arguments are pretty good. They're not absolute. They're certainly not certain, right? The thalamus may integrate, however, frontal cortex and basal ganglia, which do underlie the ability to modulate behavior, mediate motivation, and emotional drive. This has been shown via lesions, right? And a lot of course, subclinical studies done in rats and mice, mutations and whatnot. And they participate in planning cognition and existence of specific connections between different regions of the thalamus and limbic system, the affective region, suggesting dysregulation, which is seen, of course, in depression, right? It's part of the clinical presentation. Loss of white matter integrity found in MDD, particularly in the thalamic projection fibers, the limbic system, and the frontal cortex, also support the theory of limbic, dorsolateral, prefrontal cortex, thalamic dysfunction in unipolar major depressive disorder. The specific area of the thalamus, dorsal medial, is proved to integrate specific communication with the frontal lobes and involve the regulation of cortical networks, especially when the maintenance and temporal extension of persistent activity patterns in the frontal lobe areas are required, playing perhaps a critical role in higher cognitive functions together with the prefrontal cortex, the executive functions of the PFC. Leptin was significantly increased, okay, and we're analyzing the data, in MDD, and positively correlated with severity of the disease. Okay, that's what we saw in those histograms. Leptin and glucocorticoids play opposing roles depending on neuroendocrine environment. That's a key thing to remember. And I wanted you to take a look at the weight of leptin and immunity. This is a paper published in Nature Reviews Immunology way back in 2004. Take a look at this. So you have overeating, acute phase response. You have inflammation from interleukin-1 and TNF, and also hyperinsulinemia. Stomach and skeletal production. You have leptin going into peripheral circulation from the adipose tissue. What blocks that, or what would block that, is fasting, reduced food intake. Testosterone blocks that, so that's why 2-to-1 females have higher levels of leptin in general than male do, at least in males who produce a lot of testosterone. The adipose tissue that is producing leptin is getting into peripheral circulation. Peripheral effects, T cell function, hemopoiesis, angiogenesis, pancreatic beta cell function, that's the insulin connection, reproductive function, basal metabolism, central effects, of course, food intake, HPA, axis hormonal regulation, bone formation I talked about already. You get working through the lymph node and through the lymphatic system, the production of pro-inflammatory cytokines, IL-1, IL-6, tumor necrosis factor. More leptin can be generated directly from these immune cells. You get central nervous system and hypothalamus production of the CRH, right, corticotropin releasing hormone. This is your whole HPA axis. This is your canonical HPA axis, for those of you that know it. There's ACTH generating glucocorticoids, which then work directly on the thymus. This is linking leptin to the production of glucocorticoids. Why am I saying that? because high levels of circulating glucocorticoids is another paradigmatic serum marker for major depressive disorder. High leptin, high glucocorticoids. You see the connection? This is when it was first put in this 2004 paper from this Italian group, from Lacava and Matarizzi, Italian researchers. This was in Nature Reviews Immunology way back in 2004. So I'd like you to see where this information comes from. Okay, so here's putting it together. This is from a paper published in BBA, Molecular Basis of Disease. Now we're up to 2014. Glucocorticoids do what? They increase adipogenesis. Okay, that means more lipid produced. They increase adipocyte metabolism. They shut down glucose metabolism, so you have high circulating glucose now. They alter glucose sensitivity, either increasing or decreasing it depending on the other factors like beta cell function. And with lipid metabolism, same thing. Depending on the organ system involved, you can have increase or decrease in lipolysis. These are the enzymes involved: hormone-sensitive lipase, monoacylglycerol lipase, the AT-glycerol lipase. Okay, these are all enzymes which take fatty acids from triacylglycerol. That's what these all are. You also, however, get increases in triacylglycerol in other organ systems. So where you get lipolysis in serum circulating cells and in the liver, you can get an increase in triacylglycerol synthesis, therefore fatty acid synthesis, acetylcholine carboxylase, diacylglycerol acyltransferase, and more lipoprotein lipase, which allows you to move fatty acids around either between phospholipids, sphingolipids, and triacylglycerol. All that happening in the adipose. You also get from glucocorticoids. See how this is not, it's not giving you the excluded middle term. It's showing you that different organ systems are responding to glucocorticoids differentially, right? Adipokine function, leptin increases, right? RBP4, these are all proteins associated with that pathway. Adiponectin, depending on which system you're looking at, can go up or down. And in terms of stromal cells, you can get a decrease in some of these pro-inflammatory cytokines, depending on whether or not adiponectin is increased, right? It's just showing you the pathway with glycerol-free fatty acids. There's the lipase producing monoacylglycerol, diacylglycerol, triacylglycerol. This is adding fatty acids, storing triacylglycerol. These are all the enzymes which allow for you to acyltransferase fatty acids directly to a diacylglycerol preformed structure. Here's the kinase regulating the amount of glycerol 3-phosphate, which will give you glycerol to make these triacylglycerols, right? So you have VLDL giving you fatty acids from the lipoprotein lipase, glucose through the GLUT transporter giving you glucose, making through glycolysis, glycerol 3-phosphate giving you the glycerol backbone to make triacylglycerol in the adipocyte, okay? This is just showing you glucocorticoids can cause triacylglycerol breakdown to free fatty acid, giving you high systemic free fatty acid, which is a bad dyslipidemic system, but you also have the production of triacylglycerol because of glucocorticoids. So it depends on this rheostat control. So finally, what does all this say? Glucocorticoids, which is what GC stands for there, simultaneously increase triacylglycerol synthesis and breakdown. Glucocorticoids, especially when added to in the presence of insulin, mimic the fed state, induce the expression of genes involved in lipid uptake, like lipoprotein lipase, triacylglycerol synthesis. Those are all those enzymes. The lipins actually are involved in what? That's involved in oil droplet formation. These are the acyltransferases, okay? Glycerol 3-phosphate acyltransferase, diacylglycerol acyltransferase, de novo lipogenesis, that's acetylcholine carboxylase isoform 1, FAS, that's fatty acid synthesis. Glucocorticoids, however, also increase the expression of genes involved in triacylglycerol hydrolysis. This is the triacylglycerol lipase, hormone-sensitive lipase, and the monoacylglycerol lipases. Therefore, glucocorticoid induction of PCK1, that's a kinase expression, is likely to play a role in glycerinogenesis, that's glyceride produced from glucose, right? Glycerol 3-phosphate. that contributes to triacylglycerol synthetic pathway. That's up there. Okay. Glucocorticoids also increase lipid turnover in the adipose tissue, while glucocorticoids are widely cited as lipolytic and thought to increase systemic free fatty acid, like down here. They also increase lipid uptake and triacylglycerol synthesis. So any factor that tips this balance between triacylglycerol synthesis and adipose fat-dependent matter may actually lead to hypertrophy of existing adipocytes, increasing their mass, hypertrophy, and hence affect regional adiposity. Okay, where is that regional adiposity going to be? For example, in cardiovascular beds, right, or in solid organ tissue. All right, so it gives you more detail here. This comes from this particular site. Glucocorticoids modulate adipogenesis in various metabolic and endocrine functions. Major depressive disorder linked chronic excess glucocorticoids linked to increases in central adipose depot and contributes to dyslipidemia. The intra-abdominal adipose, which builds up, may be more responsive to glucocorticoids because of its high glucocorticoid receptor expression. So you got to remember that the receptor also modulates. So even with high circulating glucocorticoids, that's why you can have either synthesis or degradation of triacylglycerol, depending on whether or not you get to the correct pharmacokinetics and pharmacodynamics of these compounds associated with either drug delivery or drug utilization. They're trying to impact these pathways for people that are, for example, obese, whether or not you're going to get an increase or decrease in glucocorticoids and therefore mediate the leptin response, which can then lead to behavioral responses in the central nervous system associated with potential disease states like major depressive disorder. So multiple GR isoforms, those are glucocorticoid receptor isoforms, interactions of that receptor with other transcription factors in the adipocytes and mediating inflammation is still being sorted out. So that's what you're showing here. Hypothalamic reception of leptins can associate with reproduction and growth, metabolic rate we talked about, increasing actually sympathetic tone like high blood pressure. There's an immune function we talked about. We talked about the adipocytes generating the leptin. Insulin can induce that. You have counter-regulatory hormones constantly regulating at the adipocytic level. Corticoids may be enhancing this whole response. Corticosteroids being generated. Leptin resistance may be working here because of excess control. Leptin resistance then have not being mediated because excessive feeding. Glucocorticoids are distinguished from mineralocorticoids and sex steroids by their specific receptors. I want you to understand this because you need to know a little bit more about lipids. Because its receptors, target cells, and effects. A corticosteroid, as opposed to a glucocorticoid, refers to both glucocorticoids and mineralocorticoids, as both are mimics of hormones produced by the adrenal cortex. That's why they're called corticosteroids, you see. But it's often used as a synonym for glucocorticoid, but it should not be, you see. Because glucocorticoids don't include mineralocorticoids. Mineralocorticoids do all kinds of things with calcium and phosphate and selenium metabolism through the kidney and malabsorption or reabsorption, and then the urea cycle and whatnot. There's a whole lot of other biochemical pathways that are not associated with glucocorticoids that are associated with corticosteroids. Be very careful about using those terms. I'm sure from now on you will be. All right. So we're going to stop there. We're at the end of this discussion of this paper. Now, one thing I want to point out to you, okay, as kind of like a prolegomena for the next chapter we're going to get into, where I now apply my diet event ontologics to looking at leptin and looking at obesity and written large all of the neuropsychiatric disorders, and then linking back into metabolism and aging and sexual dimorphism. Before we get there, I want to say one more comment about this paper, and then I'm going to do it full-blown in my next lecture. Remember that all the people, medical associated naive major depressive disorder patients, major depressive disorders that were on an SSRI or an SNRI or perhaps a glutamatergic pathway inhibitor or activator or agonist, thus modulating depression, or even the healthy controls. Remember, they were all lean. Yet there were slight differences in leptin, but they weren't very large either. Remember that? Yet the paper is trying to tell you that slight elevation in leptin affected the white matter anisotropy, and they're trying to argue, very, very, very smoke and mirrors argue, I would argue, that means that naive MDD people, high circulating leptin, high pro-inflammatory responses, thus giving you the presentation of more severe major depressive disorder according to the scales used, the dynamic scales used, the instruments used, than people that are medicated who have major depressive disorder as compared to the healthy control. I would say that the whole argument, in a way, collapses onto itself, moria sua in Latin, collapses upon itself, because none of them were obese. So what would induce the high levels of leptin production? Because we're telling you that leptin is a chemical rheostat coming from visceral fat. So just because the leptin levels were slightly higher in the drug-naive MDD patients, and therefore they're arguing that had an effect on their depression, remember that the people that were even on the pharmaceuticals, they're supposedly then having some kind of remission of depression, that their scores on the Hamilton were not so great. That means that this whole argument about leptin mediating white track anisotropy, which is what they were measuring here. Remember, they did all that DTI study. I wasn't too impressed with it, to be honest with you. That particular, the data wasn't, even though the statistics seemed to suggest there were differences, remember. P-values weren't great, also remember that. So my point is this. A lot of papers are published that are well-intentioned. The authors seem to have a pretty good understanding of at least one thing. I think these people did not really understand lipid metabolism. They did understand white track mediated major depressive disorder. They wanted to see if leptin played a role. They did this pilot study that looks like they think that they did find hyperleptinemia associated with increases in major depressive disorder. They said, therefore, that's linked to this white track anisotropy. And I'm saying that's where they made the mistake. They made too far of a leap of faith, an inductive move from the data. Okay, and if you make that kind of movement, that really huge movement, there's a paralogism. So paralogism is going beyond what the premises will allow to come to a conclusion. Okay, and because they made that mistake, a paralogistic mistake, I think that you cannot say, because of the publication of this paper, that otherwise was done well, that high levels of leptin regulate or map onto, because I want to say regulate because that's too strong of a word, map onto anisotropy of white tracks, which maps onto major depressive disorder. You cannot make those links. There's big question marks there. And so I'll leave you with that. I want you to think about that because that's really important in understanding whether or not you can take a published paper like this, right? You can take a published paper that we're looking at and say, oh, yes, indeed, they have proven or they have good data that represent that leptin and obesity could cause depression, because that's where they're leading to. They're saying that obesity can cause depression, not that depression causes obesity, right? So the difference between what? Empirical, right? Empirical going from thinking to being, or reasoning going from being to thinking, relative to ontic factors or events in the world, right? Again, dealing with principles of logic to look at how data is represented and how the thought process of the researchers comes through. So with all of that in mind, I'm going to say goodbye for now, and we'll catch with you later when we do the diet event ontology of this really interesting work. And other papers will then, of course, be brought into play. Thank you for your patience. Have a good afternoon. Dr. Dan J.Guerra saying bye for now.